So I'm here to talk to you about something very different and different from the data perhaps you've seen before. I'm going to talk about brain data, and specifically the time-resolved brain data, and I'll tell you what it is. And I'm going to give away my main message of the talk now is that, firstly, we know very little about it. Secondly, we don't yet know how to analyze it properly. Thirdly, this will tell us how the brain works eventually, if we get it right. Oops, no, that wasn't the click. And thirdly, it has a huge practical implications about solving and understanding how do the developmental brain atypicalities or disorders come about, and potentially what the brain does to solve them inside itself, and what eventually down the line, what policies we should put in place, what can it tell us about what we should be doing. Right. So this is a huge topic, but what is time-resolved brain data? Click. There you go. What we normally think about as brain data is MRIs. Maybe some of you have been in MRI. When it's a big noisy machine, you lie down and you get a picture of your brain. Looks like that. No, mostly you standardified huge or either macro or microstructural abnormalities of the brain tissue. Not just the brain tissue, but also, of course, the skull and all the other tissues inside your head. But that's the only thing that can do it. It can do many more things. And one of these things is to understand where the blood flows in your brain. Right. So we've been using this data to, let's say we give you one task, maybe a reaction time based task, or you're clicking, you're seeing pictures of cats or dogs. And then we give you the other task and we trace how the blood flows through your brain. And by contrasting different tasks, we say, oh, in this task, you have more blood in these brain areas. Now, blood is slow. Blood is not the direct measure of brain activity, it's a secondary. Your brain uses oxygen in your blood to feed itself, if you like, to feed the activity. But that's not the activity itself. So you can only really measure this sluggish, what we call hemodynamic response over, let's say, 7 minutes. So you're really averaging over a huge amount of activity and saying, what it does tell us is this sort of brain areas are involved in these sort of tasks. It's not going to tell you how it does it, right? Because that's very slow response. The brain works on millisecond scale. This is minutes. So, yeah. Thank you. So what I work with is things like this. This is called MEG and EEG. So the one on the left is MEG and the one on the right is the cap, the EEG. They stand for magneto and electroencephalography. And what they measure are brain waves. So this is a direct measure of neural activity. The neurons fire together in mass ensembles, and we measure either the electric or magnetic flow of this activity over your skull. You get sensors either in that dome of the MEG or in the cap that we place on your head, and we sort of try to understand millisecond by millisecond what your brain is doing. So if I say, hello, my name is Anna, that's what, 700 milliseconds a second? That's very quick. But your brain will analyze that at that speed and probably will also think about something else as well in the background. But what we can get out of this data is, if we look at that little block of data, this would be amplitudes on the vertical scale and the time on the horizontal axis. And this is individual sensors. So I'm collecting the data for each of the sensors here. And this is the perfect snapshot of what is happening in your brain whilst you're hearing that sentence, presumably analyzing it. So somewhere in that little square of data, little matrix of data, is what you're hearing and how you're processing it. Now, we don't know how to decode this, but we really try hard. And this is a wild west of data analysis of how to get information out of it. I will tell you very little about how to do this. This is a hard problem. Anybody who wants to solve really hard problems are welcome to work on this. But I will tell you how we can use it and maybe some more simple ways that we can analyze it. Click. So I'm going to focus on the example of dyslexia today, because this is the work I do. But this can be applied to many other developmental atypicalities. The thing with developmental disorders or atypical trajectories is that they're not always embedded in brain anatomy. So when you have MRI, you have a brain scan. If you have a tumor, oh, there it is. When you have difficult atypical development, this either the differences are very subtle and microstructural and very different person to person, but what will tell you what's happening is the brain activity. You will analyze information differently if you have developmental atypicality. Not necessarily worse, sometimes better, and this is something I will show you today as well, but you will do it differently. But that data is hidden in the time-resolved brain activity, not in the brain picture. So how do we know that? So dyslexia is, if you read all the papers in the world about dyslexia and try to summarize them, it will tell you something like dyslexia is a phonological disorder. Basically, it's a difficulty mapping the sounds to letters, right? If I tell you b, you know how to spell it. If I tell you k, you will write it down for me. And children that's been diagnosed with dyslexia, they struggle with this. And then this has a massive knockoff on their reading, because the first time we learn to read, we look at the letters and we one by one string them together, or syllable by syllable, and we try to map them to words. Now they struggle with that, but where does this come from? We know it's a phonological disorder. Where does this come from? There are many theories out there. The one I am currently working with is being proposed some time ago. And the idea is that it comes from very early in life. It comes from the fact that you're struggling to trace the speech with your brain. What I mean by this. So if I say again, hello, my name is, what if you record me and then you try to plot the acoustic energy, it will look like something like this. And these peaks represent typically the onsets of salient events in the speech stream, the onsets of phrases, words, syllables, pockets of information that your brain takes and decodes. But to understand where these pockets of information lie, your brain needs to learn to attune itself to this information. How does this happen? Well, we call this speech to brain entrainment. So somewhere in your brain, how this works, again, we don't know, but this is what happens. You're listening to me and your brain starts to oscillate at the rhythm of my speech. Why does it do it? Well, so if you pretend that this is high frequency squiggly line is my speech, and this top, the less, the low frequency line on the top is your brain, it basically traces the peaks of my voice over the acoustic energy to create in itself pockets of information analysis. So the brain goes up when there is important information to be decoded. So this is very simplistic understanding of what it is, and we still don't understand what it is. But this is what we see when we analyze time-resolved brain data. The brain goes up when the acoustic energy goes up. Brain goes down when the acoustic energy goes down. And the idea is that this helps to decode the speech, segment the chunks of information. What should I analyze? And another important part is we know that dyslexic individuals don't do it quite as well. In fact, there is some amazing work being done now with neonates showing that those with a history of dyslexia don't trace their parents' voice quite as well with this rudimentary, very slow brain waves. And then developmental studies starting to show that this strongly correlates with their reading skills later, how well they read individual words. Now, the fun thing is, despite of this issue, they still go on to develop typical speech processing. They still understand speech. They can communicate. It's when the reading starts, that's when the issues start to begin. And this is one of the big questions as to why, if they don't do this quite as well, how come they learn individual words? How come they segment the speech in a different way? Perhaps they still get to more or less okay speech comprehension. This reading, this the issue. And this is one of the questions that we pose in my work. So what can the brain resolve data? So this already told us there's an issue with tuning the brain to the speech signal. But what more can it tell us? Because in that brain activity is encoded not just speech, not just understanding of individual sounds of speech. Speech is also context, right? If I tell you the dog is chasing the cat, you're not surprised. the dog is chasing the chicken, not surprised. Maybe a little bit more surprised. Dog is chasing an elephant, very specific context, surprise. So what typically happens is that you're trying to guess the upcoming words. You're integrating words in the sentences, but you're doing so predictably. And the words that are more predictable, if I said the dog is chasing them, most of you already finished, like, I know it's a cat, right? It doesn't need to tell me that's a cat. And we see the brain does that by sort of reacting more to the words that are less predictable. So the higher the probability, the more reaction there would be in the brain. But we could also measure objectively how predictable the individual words are in the sentences with some neural networks that are designed to guess what the next word is. So we can map saying, okay, for each of these words, we can map how probable the sentence is. The fun thing is the brain traces that information as well. As much as it traces the contours of speech, it also traces the contours of context, how predictable words are. So it's trying as hard to anticipate what's happening. So we ask the questions, all right, if dyslexia is worse in tracing the words, the signal itself, is it worse also tracing the secondaries of this signal? So the information encoded in this, namely the probabilities of words that represents how well you're semantically analyzing the sentence. And the answer is, well, let's start with the beginning. So this, what I'm going to show you now is the little our little brain collected with the EMG. This is summary of activity over time, how well they're tracing the envelope of speech. And the blue is dyslexic, the blue is control, the red is dyslexic data. And the higher the colors are, the more vivid the colors are, the better the tracing of this information in the brain. We do it with a complex techniques, multivariate techniques. I'm happy to talk about them later. No time now, but ask me how. And we can see, maybe not with a slide, but the controls are better at this. This is something that we already know. The important part is the better your brains are tracing this acoustics of speech, the better your pseudo-word reading, the phonetic decoding of speech. This is when you read fake words and you have to read them letter by letter, essentially, to understand what the sound is. And this is true for everybody, not just for dyslexic participants. So the dyslexics are somewhere here on the bottom of the graph, but that's true for everyone. So this is very important. This is true for all the individuals. But the fun part, what we found here, is that when we look, instead of tracing the sound, tracing the content and the context, how well you analyze in the context, they're the same. They're not worse. They're the same. But even the funnest part is that the better the dyslexic individual are tracing context, the better that their reading is. So that screams a compensatory strategy to me, right? So the better you are at understanding that it would be the cat that the dog is chasing, the more is in speech, the better your reading will be down the line. So it seems to me that this is the mechanism that is sort of, they know that they can't do the top bit. They know they can't trace the acoustics very well, but maybe they can compensate a little bit by the context. Not so for controls. No relationship, because they learn to read in a different way. They learn to read with phonological decoding. And this is just a little bit of what the time-resolved brain data, tracing speech in the brain, can tell us. So summing up, I probably overrun. Time-resolved brain data is a powerful and very rich source of information. It also allows us to test people in a naturalistic setting. We're not sitting there with a pen and paper test. They're just listening to speech, audiobooks, movies, and then we can just analyze what they do in the brain. For dyslexia, able to understand the neural mechanisms, so the tracing of different parts of information, and also give us the clues about potential ways that the brain tries to compensate itself. So maybe we can understand how brain does it and then apply that, help it later down with some policies. And this is it. Thank you very much.